Guys are going to get started. Just a heads up, they gave us more pizza than we ordered, so if anybody would really like to take a pie home for dinner, come and see us at the end. Okay, so we are the Center for Cardiovascular Investigations at the University of Guelph, and as most of you know, we represent faculty and colleges all across the university, and so you can see them listed here as well as the faculty members. And the basic big idea of those of us who came together and started in the first place was we really wanted to transform our thinking about cardiovascular disease. We wanted to be able to extend our capabilities so we could do everything from basic research to clinical translation, and that's both in animals and humans. And the basic idea was to discover new ways to diagnose heart disease, advance therapies, and train the next generation of clinicians and scientists. So some of you have probably seen this slide before, but it's a great area of research to be involved in. Cardiovascular disease is our leading cause of death, and so lots of effort goes into sort of trying to find cures, but also decrease the morbidity and extend it, help people live longer, healthier lives. And so we have been awarded over $14 million in competitive research funding collectively over the last 5 years, and that includes every single major funding agency in Canada. So if you want to know more about us and more about what we do, we have a webpage. If you go Cardiovascular Research Guelph, it'll come up. You can see about the investigators, students, educational programs, things like this that we run, facilities. We've updated everybody's publications from 2018. If yours is missing, just email us and let us know, and we'll make sure it's added. If your picture's not on there, if you're a member lab, then just let us know and we'll add you on there. And then university policies. So we do a number of different types of talks, and this one is our Cardiovascular Scientist Seminar Series, and we have three reasons for doing this. The first is that we want to promote the outstanding research that our faculty do. Most of us are getting invited to go talk somewhere else, and we never hear what we do, and so this is an opportunity to hear about our research. Second reason is for the graduate students so that we can collectively see what the other labs are doing and create collaborations and build capacities with our research programs. And the third is the undergrads who want to get involved in research and want to get into labs have the opportunity to start to come and see what people do, and then they can talk to the researchers if this is types of research they want to do. So we have different themes, and we try to kind of pair animal and human. And these are ones that have been going on over the past few years. We also do our Distinguished Scientist seminars, and if anyone has a Distinguished Scientist, these are one-ofs. We can usually do 1 or 2 a year, but feel free to reach out to the executive. Let us know, because we can bring down people, or if you've got people here for PhD defenses and stuff. And the third thing we do biannually, every other year, is the Cardiovascular Research Day. So that's all I'm going to say. I'm going to call up student executives to chair now.
Hi everybody. On behalf of the Student Executive Council, we welcome you to the Center for Cardiovascular Investigation Cardiovascular Scientific Seminar. Just a few notes before we start. First, take a photo, tweet photos, and use hashtag CCVI, hashtag Guelph. You are also welcome to follow us on Twitter and LinkedIn. Next, a special thanks to all the members of the Student Executive Council and all undergrad reps for helping to coordinate their labs and promoting today's event.
So we're now going to call up Dr. John Dawson just to introduce Right for Heart.
Thanks. I'm calling up Grace just to embarrass her as well. She doesn't want to speak, but this is Grace. We are the co-captains for the Ride for Heart team from the Guelph Cardiovascular Center. This is also a wonderful opportunity for us here at Guelph to showcase that we have this wonderful group here, because we go to Toronto, we ride our bikes, or we go for our walks, and we are in a VIP team, which means we have a table and so on inside the pavilion. It's a massive event. But our table is right beside the guys from Toronto, from Hamilton, and we get to talk to other researchers, and it's also an opportunity for us to put a more public face on the Guelph research that's going on, and it's an opportunity too to give back to the heart and stroke. So we're putting together that team. Grace and I are the co-captains. You're going to be hearing more from us in the coming weeks as we start to ramp up fundraising here at Guelph and also getting people involved to sign up for that. We have a code to give you a 50% off, right, on the registration. So if you know how to ride a bike or know how to walk, you're eligible. And you can just go and do that. So let us know. Grace's email is on the bottom there. If you're interested, please get in touch and we'll tell you more as time goes on. Thanks, everyone. Yep.
Okay, so we're going to transition into our talks for today now. So just a few housekeeping notes before we start. So we're going to ask that everyone remains for both speakers. We're going to hold all questions until the end when both speakers are done, and then you can just come down to the front and ask any questions. And so we're going to do it that way so we can hold the talks in the time permitted. So our speakers, you have 20 minutes for your talks. So try and stick to your time. Our student reps will come down to the front if you're running over to encourage you to wrap it up. And lastly, we want to thank Biomed, OVC, and CBS for sponsoring today's pizza lunch. So we now have the honor of calling up Dr. Francesco Leri, who's the Associate Dean of Research and Graduate Studies for CSAHS, and he's going to introduce our first speaker.
Thank you. Not deserved the clapping. I appreciate it, of course, to participate in this and learn about the research. So Dr. Todd Gillis is an associate professor in the Department of Integrative Biology at the University of Guelph and the founding executive faculty member of the Cardiovascular Center. He did his graduate training at the University of Guelph and Simon Fraser University, and then worked as an NSERC postdoctoral fellow in the School of Medicine at the University of Washington in Seattle. During this time, Dr. Gillis also visited the McMurdo Research Station in Antarctica to study the hearts of Antarctic fish. Dr. Gillis was recruited to the University of Guelph in 2005. The work in his lab is focused on the molecular and cellular mechanisms responsible for regulating heart functions and how this change in response to physiological and pathological stressors. The result of this work are increasing our understanding of the mechanisms that regulate and limit the adaptive capacity of the vertebral heart, as well as identifying strategies that could be used to help treat human pathologies. Dr. Gillis has recently published 2 new books. One is The Cardiovascular System, Morphology, Control and Function, and the other one is The Cardiovascular System, Development, Plasticity, and the Physiological Responses. The title of his talk is How to Function in the Cold or Without Oxygen, Novel Things Learned from Studying Fish Hearts.
Okay, thanks for that introduction. How's my voice? It's all right? Okay. So here's the title of the talk I submitted. Here's actually what I'm going to talk about. Because of time, I would have to speak a lot faster than I normally do. And so what I have up here is a slide, and these are animals that we're currently working on. We've published papers on recently, or we have something in submission. And my lab is a comparative cardiovascular physiology lab. We work on different animals, and we're interested in the function of different hearts, because I think these provide a lot of interesting insights into the capacity of the vertebrate heart to adapt to different physiological conditions. We've just submitted a paper that's looking at developmental programming in alligator hearts in response to hypoxia using a proteomic approach. We have done a lot of work with wild salmon, sockeye salmon on the west coast, and we're looking at how their hearts are affected by bitumen exposure, which would happen if we got a rupture of the Trans-Canada Mountain Pipeline. Done a number of papers working with John Dawson in the CCVI, looking at zebrafish and how their hearts are regulated during development and how that can be affected by the expression of transgenic proteins. We've also done work on the diaphragms of mice, and this is with Dr. Jeremy Simpson. We've published a couple papers looking at the influence of heart failure on diaphragm function. I was going to talk about the hagfish. This is an animal that lives at the bottom of the ocean. It eats dead things, and it can survive about 36 hours without oxygen. And we've now published a couple papers working with Glenn Pyle, looking at how the heart of that animal is able to continue working without oxygen. I encourage you to go look at that paper. One just came out, I think, 2 weeks ago. Today I'm going to talk about the lowly trout. We've been looking at this fish for a number of years for some interesting reasons. And so the title of this is Temperature-Induced Cardiac Remodeling in Trout. This has been the work of a number of my graduate students. It was started off by Jordan Clayman, who defended about 4 years ago. Current student Elizabeth Johnson is about to submit her thesis, and then Dr. Courtney Deck did some work on this as an undergraduate. This has also been a collaboration with Glenn Pyle and Dr. Sarah Alderman. And so kind of referring back to that picture at the very beginning where all the pictures of the people within the CCVI, I think the work that we do in my lab is a good example of how the integration of people with different skills and approaches can work together to ask some interesting questions. And so that's kind of also what I wanted to emphasize with this talk, because we work with each other. Okay, so what's interesting about trout? They live in temperatures, in temperate conditions, so around here. In the summertime, they get up to about 20 degrees, and in the wintertime, they get down to about 4 degrees. And what's interesting is that the heart keeps working over that range of temperatures. They remain active. They actually reproduce in the springtime when the waters are very cold, and they're quite active. So that means their heart is able to work. And that's interesting, because if I was to take your heart out, decrease it by 10 degrees in temperature, it would stop working. It couldn't generate as much force, and it couldn't generate force as quickly. So the ability to pump blood would be dramatically affected. And that's because of the effects of temperature on the calcium sensitivity of the preparation. This is just a line I've made up, but it's supposed to represent force being generated at 37 degrees in response to calcium concentration. And it increases and reaches a maximum at about 5 micromoles. And this is how your heart's working right now. If I was to decrease its temperature by 10 degrees, this curve would shift to the right. The heart's becoming less sensitive to calcium. More calcium is required to generate the same amount of force. And if I superimpose on this, the calcium concentrations that are actually within your heart, you see there's a dramatic consequence. This is where your maximum force generation is. You reduce by 10% or 10 degrees, it drops down dramatically. So the heart becomes non-functioning. So this is something that the fish heart is basically able to compensate for by modifications it makes seasonally. As that animal's body temperature is changing from 20 degrees in the summer to 5 degrees in the wintertime. So something's going on. And we've looked at this using a number of different ways, and I'm going to summarize that work today. I'm going to talk about work that we've done looking at the cardiac contractile function, and then I'll merge in the stuff we've been looking at in terms of the composition. We also have done a lot of work on the anatomy, but I'll focus on these two parts today. So, cardiac contractile function. This is basically describing the ability of that heart to contract, and we use two different ways to characterize that. One is a skin myofilament preparation. This is about 1 millimeter long by about 200 microns. And we dissect these out of the hearts. We wrap the ends of them with tin foil, these little clips that we make, and then we activate them with calcium, and we look at their ability to generate force in response to calcium. This is the skin preparation. We're getting rid of the membrane, so we're only looking at the contractile function of those myofilament proteins. Another preparation that we've developed in collaboration with Glenn Pyle is something called a modified Langendorf prep, where this is a balloon that we insert into the ventricle. It's attached to a hollow tube which is connected to a pressure transducer. And that gives us the ability to measure the ability of that heart to contract and generate force. We perfuse it, and then it's also activated electrically to stimulate contraction. And we can do these preparations for long periods of time. For the hagfish work, we've done this for 16 hours of time without oxygen, but I can't go there today. So, thermal acclimation, that basically means you're taking an animal and you're acclimating it to a different temperature. And one of the protocols that we use is we keep our fish at about 12 degrees. They're in a tank. We have a second tank that we will move up to 17 degrees, and we'll keep that group of fish at that temperature for about 8 weeks. And then we would have a third tank of fish that we move down to 4 degrees, keep them there for 8 weeks, and then we look at their hearts. We want to see what's happening to the function of those hearts during those thermal acclimation periods. And so this is data from this force measurement experiment where we're looking at the function of that really small piece of heart muscle. And on the x-axis is calcium concentration, and it's increasing from left to right. And I'm showing it here as a PCA, which is the negative log of the calcium concentration. So the smaller the number, the higher the calcium concentration. Just think of it as calcium is increasing. But what you're seeing here is force being generated in response to that calcium concentration as we're basically exposing the preparation to more and more calcium. And I've color-coded the lines here, and these experiments were done at the exact same temperature. So we have trout that were acclimated to 4 degrees, to 12 degrees, and to 17. And what you see is that this blue line is shifted significantly to the left of the black and the red. And that means that this heart and this heart muscle has become sensitized to calcium. Somehow, through that cold acclimation, changes have occurred in that myofilament that now allows it to bind calcium at a lower calcium concentration and generate force. And this helps to compensate for the desensitizing effect of low temperature on heart function. So this is the myofilament work, and we want to see how this translates into the functioning of the intact heart. I'm not going to show you all the data, but this is some important data, and I'm just going to have you look at this top line here. So here we have the heart. There's a balloon in the ventricle that's attached to a pressure transducer, and we're able to measure the pressure being generated and how fast it's being generated. And this is a measure of dBdt, which is the change in pressure per unit time. And on the x-axis is the volume of that balloon. We want to see if you stretch that ventricle, do you get more force being generated? And we see that. But what's important is to compare at the same balloon volume, the blue versus the black versus the red. And this gives you an idea to the ability of the heart to generate power. And you see that the blue dots are significantly higher than what the black or the red. We've done all the required statistical analysis, and in fact, this is a statistically significant difference in the capacity of these hearts, when they're intact, to generate power. So that means that the level of the heart, it's essentially stronger. It's able to generate more force in response to calcium. So there is a real change in the function of that heart as a result of that 6 to 8 weeks of thermal acclimation. And so we wanted to know, what is the mechanism for this? We're quite interested in the proteins, how they work with each other, how you can change them to alter cardiac function. So we've done a variety of things working on this, and one of the things we looked at was protein phosphorylation. This is a mechanism by which you can regulate cardiac function relatively quickly by adding a phosphate group onto proteins. And one thing that we've noticed is that there's basically a standard decrease in the level of protein phosphorylation in these cold acclimated animals, and that the decrease in the level of phosphorylation for cardiac troponin T was significantly decreased. We've also found changes in troponin I expression, but I won't talk about that today. So this suggests that these animals are modifying their myofilament by phosphorylating proteins and changing the proteins that are actually there. And again, that's all within that 6 to 8 week time period. So summarizing this first part, cold acclimation increases the level and rate of pressure generation. Increased pressure generation, we think, is as a result of that increased calcium sensitivity of the myocardium. That was demonstrated by the myofilament experiment. And we think that this increased calcium sensitivity is due to manipulations of the myofilament, protein phosphorylation, and changes in the myofilament complement. The next thing I want to talk about, and I'll talk a little bit more longer about this, is changes in the actual heart structure and its composition. And I'm going to focus in on the work that we've done with the composition. So hearts are made out of muscle, primarily, and connective tissue, and that affects the biomechanical properties of the heart, the ability of the heart to fill with blood and then to contract during systole. This is a picture of a trout heart, and quite different from ours. It's not a hollow chamber that fills with blood. Instead, there's these channels called lacunae, which insert into the spongy myocardium, which is being shown here. This is all muscle tissue, and it contracts as our hearts do, and it is made up of the same protein complement and works the same way as our hearts do. And the protein sequences of these proteins are actually quite similar between all vertebrates. And so this is work that we did following a similar thermal acclimation, and we stained these hearts using Mason's trichome, which stains muscle red or pink, and it stains collagen blue or purple. These are the fish that were at 11, 4, or 21 degrees. Looking at them, you can see that there's a bit of a change in the color. You're essentially seeing more purple on the left and more pink on the right, and this indicates that there's changes in the collagen composition. This suggests that these animals are able to modify, as a result of 6 or 8 weeks of a thermal acclimation, the amount of collagen in their heart. which is quite a novel finding. Once collagen is in our hearts, it's there typically until we die. If we get myocardial infarction, that scar forms, the collagen becomes a scar, or it's partly collagen, and that is there until the individual will die. These fish appear to have the ability to modify that. If we do some image analysis and get some numbers on that, you do see some significant differences. And what's really interesting is that the response that we're seeing is primarily within the male fish. The size of the black bar is indicative of the amount of connective tissue in the spongy layer, and it goes up significantly with cold acclimation, and it goes down significantly with warm acclimation. So these animals, again, are titrating the amount of connective tissue within that heart. And so we are very interested in that. How are these animals able to do that? How can they physically remove collagen from their heart? This is something that has not previously been identified in vertebrates under a physiological condition. Before we go there, I want to provide some background on how collagen is regulated. Essentially, you have a collagen gene. It gets made into monomers, gets constructed into fibrils, which then are constructed within the extracellular matrix into fibers. So these is a protein network that's exterior to the cardiac myocytes. So if you want to make more collagen fibers, you can basically just make more collagen. Another way that the tissue regulates collagen is through the expression of two other types of proteins. Ones are called matrix metalloproteinases. These are enzymes that break collagen down, and you think of them as Pac-Man. You make more of those, you have more of these proteins circulating and breaking collagen down. But all collagen is under very strict regulation within any tissues, and the break to, or the control of this activity is through the expression of a second type of protein called tissue inhibitor of metalloproteinases. And these basically just act to block those Pac-Men, and they can't function. So then as a result of this, you take those out of function, you can increase collagen. So we're interested in how these different components, how the collagen, how the MMPs, how the TIMPs are affected during thermal acclimation. And so we've come up with a proposed pathway. And I'm going to kind of start at the bottom, or at the top, go to the bottom, which is the response that we see, and then we're going to start talking about how we've tried to fill in the middle. So we think, well, with a decrease in temperature, there's going to be a number of steps that we're trying to figure out that leads to changes in the genes that are associated with the proteins that regulate collagen deposition. And then we get that altered collagen deposition that we see with cold acclimation or with warm acclimation. We've done work and we have made connections. I'm not going to show you the data between temperature and changes in those gene levels. And I've also just showed you the data where we get altered collagen deposition. So we've made those connections. What is also known, and then what we and others have shown, that if you decrease temperature, you increase blood viscosity. Blood viscosity is determined in part by the fluidity of the erythrocyte membranes. You do an acute temperature change on blood, the phospholipids in the erythrocytes get stiffer, they become harder to compress, and the viscosity of that liquid increases. You also see that there's an increase in ventricular pressure, and I'll explain how that works in a second. So we want to be able to figure out what's going on in the middle here. So we've come up with this hypothesis of cellular deformation. So think about a fish, it's swimming around, it's relatively happy, its heart is beating, and here's the cells that are within the heart, and they're connected to each other, blood's moving through the heart. Then you take that animal and you thermally acclimate it, and with a decrease in temperature, like I said, whoops, we get an increase in pressure. And why that happens is that if you take a trout, you reduce its body temperature, its heart rate slows down. But what's important is that they maintain the amount of blood being pumped per unit time by increasing the amount of blood that goes back into the heart during diastole. And so the blood volume increases, the stroke volume of that heart increases with low temperature. So that means that it's basically you're blowing the balloon up more, and you're acting to stretch the cells that are within that heart. And so this is why this is a little bit thinner, because we're pushing it out as that heart is stretching in response to the slower heart rate. And we think, or there are stretch-gated channels within that membrane, and when you pull on them, they basically open up and allow ions in. So that's one way we think that this process is being activated. Another is that there's an increase in blood viscosity, and that's due to the effect of temperature on the membrane fluidity of the erythrocytes, like I just said. And so think about trying to pull a McDonald's milkshake through a big straw or a small straw. It's a lot easier to bring it through the larger straw because of how thick that material is. And if you increase viscosity, it's basically trying to pull thicker material through a blood vessel and through the heart. And one of the things that it does is thought to increase the activation of mechanosensitive cell receptors through shear stress. It's pushing more against those cells. And we think this also represents a way of activating the cellular processes that leads to changes in the cardiac deposited within the heart. So we blow that up a little bit more. We've got these receptors that are in the membrane that are being stretched apart, or they're being activated by shear stress. And there's proteins in the membrane and surrounding them that are associated with Gproteincoupled receptors. And this is basically a way of activating a cellular process. And these go and they phosphorylate, or add a phosphate group, to a number of other proteins. And this represents a cellular cascade, a signaling cascade. And this is, and we basically have pulled this from studying the mammalian literature. This is what happens with an increase in blood pressure within humans, and that can lead to issues of myopathy within that human heart. But in fish, we think that this is also playing a role. So we think the stretching of the heart leads to the phosphorylation of these pathways. And so going back to our hypothesis, or back to our pathway, we're getting a decrease in temperature, an increase in blood viscosity, increase in cellular deformation. You're pulling those hearts apart. It's activating mechanosensitive cellular components, the membranes, or the proteins in those membranes, that activates a MAP kinase pathway. These are those proteins that get phosphorylated, that activates transcriptional factors that then lead to those changes in the gene expression patterns that I've previously talked about. And then that leads to the altered collagen deposition. I'm going to talk today about how we basically have been dissecting this pathway. I'm going to focus in on the connection between cellular deformation, these mechanosensitive cellular components, and the P38 junk or ERK pathway and how this can become activated. Okay. And these are, like I said, called MAP kinases. So the question that we asked are MAP kinases phosphorylated in response to trout cardiac fibroblast deformation. Why we're focusing in on fibroblasts are that these are the cells that are responsible for regulating collagen within the heart. They make collagen and they can also break it down. So we want to know, if you pull on them, can we affect their ability or their function in terms of regulating collagen? And this has been a massive part of Elizabeth Johnson's PhD thesis. She's been responsible for initiating cell cultures within my lab, which we've never done before, and she's been doing experiments on them. So she's established primary cultures of cell fibroblasts, which we now have in our freezer we can work on. This is showing a plate with these cells on it. We've done all the required antibodies to establish that we do have a fibroblast population. And one of the things that she's done in order to look at the effects of stretch is grow them on the bottom of a cell culture plate, which is a rubber membrane, and that allows us to stretch it. So we took these cells, they went to confluence, so they're all basically talking to each other. They're on top of this loading post. This gets loaded into our instrument. A vacuum gets applied, and that basically causes that membrane to be pulled down over that post, and that stretches the membrane that's on top of the post. And as a result, we're stretching these myocytes. This rigged the instrument, so we're going at about a 10% stretch of the muscle, which is physiological, and she had it going at 20 hertz, which is basically the heart rate of a fish. So now we're basically pulling on these myocytes, or on these fibroblasts, to see how they respond to that biomechanical stretch. So, and then she sampled them at different periods and looked at a number of things. And one thing we were looking at is whether or not these MAP kinases were being affected. This is looking at 20 minutes, and this is the P38, one of those proteins we've hypothesized. And you can see that there's an increase in its phosphorylation by about 4-fold after 20 minutes. Another one we looked at is the PERK, or the ERK, and you do not see a significant increase in its level of phosphorylation at 20 minutes. And this was done using Western blotting. We then went in at 24 hours, and we saw a similar increase in the level of phosphorylation of one of those proteins. Whoops, that wasn't supposed to happen. And an increase in the level of ERK, meaning that both of the molecules that we think are important for regulating that response are being activated by this stretch of those fibroblasts. So I think we're onto something. Okay, so now we've made the connection between cellular deformation, P38 junk pathway, and we think it's through this mechanosensitive cell components. We're pulling on them and initiating those cellular pathways. Okay, so one thing I haven't talked about is TGFbeta. TGFbeta is a cytokine that is quite common, and it serves a number of roles. So one thing that it does do is initiate fibrosis in the heart. What happens is if you pull on myocytes, TGF-beta increases, and then it acts upon the underlying fibroblasts to begin making collagen, and you get a dramatic increase in collagen deposition. And this is one of the pathways that gets activated during cardiac fibrosis in humans with a disease condition. So we think that this may also be relevant. And so we want to know whether an increase in TGF-beta that would occur by us pulling on myocytes or would be occurring in a heart during thermal acclamation alters these same pathways that are associated with collagen deposition. So we're going back to our cultured cell model, and we're asking the question, does the TGF-beta 1 stimulate collagen deposition in trout cardiac fibroblasts? This is looking at some of the gene transcription that we've worked on using quantitative PCR. We've looked at it at multiple time points, so exposing them to TGF-beta, we're using a physiological level, and we're seeing changes that would support a change in collagen deposition. We're getting, in black is the TGF treatment, we're getting an increase in TIMP expression that would inhibit activation. We do interestingly get an increase in MMP at 24 hours, but by 48 hours you have a significant reduction in the amount of the gene for MMP2, and by 72 hours you have an increase of the gene that would make the actual collagen. And so this suggests that there is a change in collagen. And if we go in and actually measure it using a Western blot, we see that after 1 week you have a significant increase of collagen type 1 within the heart. So that addition of the TGF-beta does cause there to be an increase in the deposition of collagen into the heart. So we now have another mechanism by which these animals are manipulating collagen within the heart. So that's TGF-beta going through this pathway. We've increased it, we've changed and got the required changes in gene expression, and now we've gone into an altered collagen deposition. Okay, this is interesting, and we found what's happening within cells, but what's happening within these fibroblasts may or may not be what's happening in the heart. So now we're doing experiments using live animals, and that's work that's being done by a master's student called Sophie Ding in the lab. And that has been going on for about the last year, and we're now beginning to get some of those results. But stay tuned for that. So being able to reduce collagen or increase collagen in the heart is interesting. We're also interested in how collagen may decrease during warm acclimation. This is what could potentially be applicable to regulating collagen in the heart of a patient following myocardial infarction. And we focused in on something called microRNA 29 beta. MicroMRNAs are small non-coding RNAs that bind to the 3' untranslated region of mRNA, and they basically knock down its expression. It takes that gene out of circulation, and as a result, that leads to there being a decrease in the associated protein. And studies with humans and with other mammals find that microMRNA 29 beta decreases in hearts following myocardial infarction, and that leads to the fibrosis. And that reason for that is because it targets the collagen transcripts that translate into making the collagen protein. So you get rid of microMR 29 beta in an infarcted heart. The response is to get more collagen. So we think the opposite is happening within cardiac fibroblasts within trouts, or that is an idea. And so we've gone back. Well, before I go there, one thing that's important is to look at the nucleotide sequence of the microMRNAs from different species, and there's complete conservation between what we see in the rainbow trout, humans, rhesus, monkeys, mice, rats, all the way down into other mammals. This means that this conservation of sequence implies importance. This sequence has been basically maintained through hundreds of millions of years of evolution, and that helps suggest that it does play a critical role in the regulation of collagen within the heart. So we've gone back to our model with the fibroblasts. We've transfected them so they make the microMR 29 beta, which is what we're seeing right here. This is a gene. And we see that after a week of following exposure, of following transfection, we do get a knockdown of the gene for collagen 1A3, which is one of the critical components for cardiac collagen. We go and we look at the collagen protein, and we see that that treatment also decreases the amount of protein within the heart. So we think that this is possibly one of the ways by what the trout is using to decrease collagen deposition during warm acclimation. And so they're basically doing that by knocking out these monomers, and as a result, causing a reduction in the amount of collagen fibers. And we're pursuing that. We're wondering, does warm acclimation cause an increase in the expression of microMRNA? And what would then be responsible for regulating that expression within the trout heart? And that's work that we're getting into at the moment. And for time, I'm going to end it there and acknowledge my funding sources, an NSERC Discovery Grant, as well as NSERC Accelerator Supplement, which funded all of this work. So thank you very much.